Everyone to the Precision Medicine seminar series. So it is my pleasure to introduce our local speaker today. It's Dr. Zhonghua Liu, who is an assistant professor of biostatistics in the Department of Biostatistics here at Mailman School of Public Health at Columbia. Zhonghua trained at Harvard in Boston. He earned his doctorate over there and then moved on to the University of Hong Kong, where he was an assistant professor with tenure for a number of years. And he was successfully recruited to Columbia in 2022. So just, little over a year ago. Where he started his research program as an assistant professor in the Department of Biostatistics. He is interested in causal inference, and clearly he will, as reflected by his talk today, by the title of his talk, he'll talk about Mendelian randomization, but he's also broadly interested in the application of statistical genetics and statistical genomics methods to problems in human genetics. And also has background in deep learning. So we are very much looking forward to learning about your latest research. Thanks for attending the seminar.
Yeah, thank you. Thank you very much for the kind introduction. It's truly my honor to be invited here to speak about our recent research results on Mendelian randomization. So thank you for the leadership of Precision Medicine Resource for inviting me. I'm so happy to be here. Without further ado, I will start my talk. So today my talk is about robust Mendelian randomization coupled with AlphaFold2 for drug target discovery. Okay, so today we have lots of data from genome, exposome, and phenome, how to figure out their relationships from genome and exposome to understand the genetic architecture and also disease etiology. That's very challenging. So both genome and exposome contribute to human phenotype, for example, complex diseases. And there may be some interactions between genome and phenome. Well, the value for big data, people have been discussing this, so-called big data analytics. So the goal is to transform data into knowledge using biostatistics, which is the work I do, and biological and medical informatics, and also domain science, people who know the field. So the goal is to use those techniques to analyze data and to discover the truth. But there's one critical component when you analyze data is how to account for randomness. That's very important when you try to infer any results from the data. So there are many examples of so-called health big data, for example, EHR data, medical records data, your genetic data, your smartphone data, lifestyle, environment, and all of this require advanced techniques from data science to, for example, figure out the causes of disease. And eventually we can do precision health, like prevention or medicine. And some part of the research will become policy, health policy. So to motivate today's talk, I want to pose this question. So proteomics data now is available in many big data cohorts. For example, UK Biobank has released more than 50,000 people's proteomics data. So the question is, if we want to figure out which proteins in the plasma they are associated with Alzheimer's disease, there's so many proteins, thousands of proteins. So the question of interest is which plasma proteins are biomarkers of Alzheimer's disease. So we try to find the link from your protein to Alzheimer's disease. And that's basically the goal we want to achieve today. So there are two types of designs in biomedical studies. One is called randomized design. The other one is non-randomized design. We might be very familiar with randomized design, for example, randomized controlled trials. We randomize people to treatment group versus control group. And the other type of design is non-randomized. For example, cohort design, case cohort and case control cross-sectional designs. Basically, two broad categories of study designs people use in biomedical research. So let's talk about the first type of design called randomized experiments, including the randomized controlled trial, or ICT. Well, this has a long history back in statistics in the 20th century, so 1923, exactly 100 years ago, by a famous statistician, Jerzy Mailman. And in 1935, Fisher, those two giant in statistics developed the series so-called the design of randomized experiments. And this idea or principle or methods have influenced modern science research, including natural science, biomedical research, and also social science research. So randomized controlled trials is one type of randomized experiments. In medical research, we randomize people into treatment group, for example, COVID vaccine, and also randomize people to control group. So here is a treatment group and a control group. And we follow up, observe their clinical outcomes, and compare the outcomes in the treatment group versus the outcome in the control group. If you observe any significant differences between the two groups, then you can claim there is treatment effects. So for example, the COVID vaccine really works by reducing mortality, okay? Or any new drug. But for some exposures, for example, this smoking, tobacco smoking, is probably not feasible to randomize people to tobacco smoking. You randomize people here to smoke and have people not to smoke. They typically won't pass RIB. So it's not ethical. It's not feasible. For many exposures, we simply cannot randomize them. If we cannot randomize them, how can we study the effects of exposure on the outcome of interest? So you cannot do randomized study here in this setting. So you cannot. So then you have to appeal to so-called non-randomized study, often called observational study. In such kind of studies, your exposure variable is not randomized. You just passively observe what's happening in the data. So I'll give you one example of observational study just to amuse you. So this study actually published in New England Journal of Medicine in 2012. Well, this paper found a positive correlation between chocolate consumption per year per person and the number of Nobel Prize laureates per 10 million people. The correlation is very high, close to 0.8. P-value is highly significant. So would you conclude if we eat more chocolates, we are more likely to get Nobel Prize? So, I have chocolate here. Who wants a chocolate, right? Keep eating chocolate, you're going to increase your chance of getting Nobel Prize. So this is a paper actually published in the medical journal, New England Journal of Medicine. You check it out. So what's going on? Based on our intuition, that's probably something not right here. So the thing is called confounding bias. Some variables may affect chocolate consumption in the country, in one country, and that variable may also affect the number of Nobel Prize laureates per 10 million population. For example, there are many possible variables. For example, GDP per capita. Okay, if a country is kind of a rich country, the education level is high. Also, the country may have more economic resources to be invested for scientific research. That's why you observe this highly positive correlation and highly significant results. So the question is, in such kind of a non-randomized study, how do we remove such kind of confounding bias? If you cannot account for confounding bias, you may get some conclusion like this, right? That conclusion can be confusing, can be misleading, right? I mean, some country may just import more chocolate and ask everybody to eat chocolate. After a few years, gonna have Nobel Prize winners in biology, in chemistry. So confounding bias is a serious concern in non-randomized or observational study. So the question is, how do we remove such confounding bias? Well, there's a gift from nature, the nature experiments. This is a biological process called meiosis. So it's a type of cell division that reduces the number of chromosomes in the parent cell by half and produce 4 gamete cells. I think I learned this from my middle school biology. It's been many years ago. So why we care about this? Because of the Mendelian law of inheritance, children, offspring's genotype is randomized. You inherit 50% from your biological father and the other 50% from your biological mother. So your genotype in this meiosis process is randomized. So that's what I say. Nature is conducting randomized experiments all the time. And how do we leverage such randomized experiments to remove confounding bias in non-randomized study when the exposure is harmful? So that's what we want to do, okay? So the idea is to use genotype as instrumental variable, or IV, because genotype ideally is randomized in the meiosis process. So this genotype can be used as an instrumental variable, or IV. We will talk about how it works. So this idea actually has a long history, back to 1928 in economics, not biostatistics or medical science, but economics in 1928. So economics people are very familiar with instrumental variable methods. For example, this lottery is randomized. How do we use lottery as a randomized variable to infer the causal effects of military service earnings, like income? There are many confounders in social science, how to get rid of the confounders. And this work, due to David Card from UC Berkeley, Joshua Angris from MIT, and Hideo Imbens from Stanford. They got Nobel Prize. Well, this is Nobel Prize. I'm not sure they eat chocolate or not, but they got Nobel Prize in economic science in 2021 for their contribution to use or develop instrumental variable approach in econ studies, especially labor economics. So in our field, biostatistics, we can use the randomized genotype as instrumental variable. We call it Mendelian randomization. Randomization because genotype is randomized, ideally. Mendelian is due to Gregor Mendel's law of inheritance in class genetics, right? So here is a cartoon to show you parents' random assortment of genes and pass on to offspring. So those genotypes are randomized, and you compare the people with mutant allele versus people with wild type allele. And you compare the results between the two groups, okay? So this is one type of randomization done by nature. Whole genome sequencing data has produced a lot of such kind of genotype data for us to be used for instrument variable studies. And this is really, we are closer to precision medicine if we have more and more whole genome sequencing data or WGS data. For each human genome, there are 3 billion base pairs, like A, G, C, G, very long sequence of pairs. And you are 99.9% genetically identical to everybody else. So what does the 0.1% account for? Here it is. In the traditional genome-wide association studies, or GWAS, people focus on common variants. As you can see here, some people with T allele, some people with a C allele. We just compare people with T allele to C allele to observe any phenotypic change. Maybe you have T allele, your blood pressure is higher, your C allele, blood pressure is lower. That's called genome-wide association studies. And the common variants, people also call it SNP, or single nucleotide polymorphism. With the whole genome sequencing data, you can also detect rare variants. Simply, it's rare in the population. For example, this position, only one person carries this A allele, all the rest G allele. So this position, this A allele is called a rare variant. Simply, very few people carry this allele. So the data type we're going to analyze called genome-wide association study summary statistics data looks like this. People run GWAS for blood pressure, for example. You have one blood pressure, you run this regression of this blood pressure for each SNP, one at a time. So you have millions of these associations. So the first column is the SNP ID, IS number, allele 1, allele 2, frequency, beta. Beta is the regression coefficients, the effect size. And SE is the standard error, sample size, chromosome, base pair positions, and last column is p-value. So this is data we're going to analyze. A good thing about this data is such kind of data are typically public available. So you can download from many websites, as we did in this paper. So for a genetic variant to be a valid instrument, there are some requirements. Not every SNP can be an instrument. It must satisfy some conditions. So the first condition we call it IV relevance assumption. So the genetic variance G here's a G, stands for genetic variance, for example, SNP. This SNP must be associated with the exposure of interest, for example, protein. If a SNP is associated with that protein, typically we call this SNP a PQTL, protein quantitative trait loci. So when you say PQTL, they mean this. Some SNP is associated with the protein. So that's the first assumption. The second assumption is IV independent assumption, which states that there's no arrow from U to G. So you don't see an arrow from U to G. It simply means there's no confounder from U to G. In other words, G is randomized in your population. Okay, in your population, or it depends on the population. So we will see this assumption might be violated in some populations. Okay. The third assumption is quite strong. It says exclusion restriction assumption. G, the SNP, affects Y, here's Y, which is the clinical outcome of interest, for example, Alzheimer disease. So G affects Y only through D. So D is like a transmission, right? It transmits G, the effect of G to Y. That's only transmission. You have only this pathway from G to D to Y. So if a SNP satisfies these three assumptions, we have three assumptions here, then this SNP is called a valid instrument. It's a good instrument. That's the instrument we want, okay? We need. So when we have these three assumptions satisfied, the goal is to estimate the causal effects of D on Y, for example, protein on Alzheimer disease. That's the goal, okay? If a protein has a non-zero causal effects on Alzheimer disease, then that protein can be used as a biomarker or drug targets. In this slide, I want to show you how Mendelian randomization works. Suppose you have G, this variable, you increase by 1 unit. So increase by 1. And you observe what's happening in the data. And you observe D, the protein, increase 2 units, okay? And you observe Y, increase by 4 units. What can you conclude from this toy example? What is the causal effects of beta? Causal effects of beta is what if D increased by 1 unit, how many units Y will increase? That's the causal effects, right? Effects always means if you increase something, what's going to happen to your Y? Okay, your outcome. So the question is, if D increase 1 unit, how many units Y will increase? And that number is called causal effects. Can you do the math? it's not a complicated math, right? So let's see. G to D, that's 2. G increased by 1, D increased by 2. And G increased by 1, Y increased by 4. And you can do the math, simply the ratio, right? So the causal effects beta can be computed as G to Y divided by G to D. And that is equal to 4 divided by 2, that's 2. So the causal effects in this example is simply equal to 2, which makes sense if you look at the figure, right? G by 1, D by 2, Y by 4. What's the number here? Should be 2, okay? So that's how MR works. But in reality, not every SNP will satisfy the 3 assumptions. It's case by case. Some SNP will satisfy the assumptions, some SNP will not satisfy the 3 assumptions. Okay? So not every SNP is created equal. Some are good SNPs, some are not so good. Okay? So there could be some violation of MR assumptions. So the first violation, what is called a near violation, is the G to D association can be weak, especially for complex traits and the diseases. So you often observe weak genetic association between a single SNP and protein. The second violation is there's an arrow from U to G due to, for example, population stratification. I will explain this concept in a minute. Third violation is called horizontal pleiotropy. So there's an arrow from G to Y, not through D. Look at this arrow here. This arrow here. Okay? When you have violations, you have to do the math in a different way. The math I just show you won't work when you have violations. Okay? So what do I mean by population stratification? This is a map in Europe. So people did so-called principal component analysis of the genotype matrix, and they found population structure in Europe. So from north to south, that's principal component 1. From west to east, that's principal component 2. And you can see a clear pattern of a population structure in Europe. Even we all call them like European population, but within European population, you still can observe population structure pattern. When you analyze European data collected from European, there's still subtle population structure. You need to take that into account. Otherwise, you're gonna bias the results, okay? So the concept of population stratification refers to there's a systematic sensory difference in your case and controls. You have case diseased people versus healthy people, and you observe a systematic sensory difference, allele differences across cases and controls. If you have that, you have population stratification. And if you don't count for population stratification, your results will be biased. You get wrong conclusion, okay? And the other concept is called genetic pleiotropy. It simply means one single gene, as we show here, a single gene is actually related to multiple traits or diseases. For example, this gene is related to blindness, liver failure, or heart, and also heart attack. It's beta globin gene mutation, single cell anemia, right? We all know this gene. And recently, FDA approved the gene editing therapy for this disease, single cell anemia. There are many types of pleiotropy. Here, I just give you a figure to show there are 6 major types of pleiotropy. So when you have pleiotropy in MR studies, you need to be careful. You need to address that violation. So the consequence due to violation of MR assumptions. So we denote this arrow from U to G as alpha-1 and G to Y as alpha-2. And we use a single notation. That's a simplified notation. We call it pi. Pi is equal to alpha-1 plus alpha-2. Now, let's review the simple math. If G is increased by 1 unit, you observe D increased by 2 units. You also observe in your data Y is increased by 4 units. The question for you is, what is the causal effects beta? Is this causal effects beta still equal to 2? Of course not. It equals to only when pi is 0. Otherwise, it's not equal to 2. But pi, we don't know pi. Pi is equal to alpha 1 plus alpha 2. What is alpha 1 from U to G? I don't know. What is the alpha 2 from G to Y not through D? I don't know. So you don't have data on alpha 1, alpha 2. So you don't know pi. Pi is called violation bias. That's the bias we don't know. If we know pi, it's simple. We just subtract off pi, we are done. But we don't know it. Once you know the bias, you can always correct for the bias. But we don't know the bias, so we can never be able to correct for the bias. So if you do the math, is 4 equal to, now 4 is equal to beta times 2 plus pi. Since pi is not a given, we don't know pi, we don't know beta. Because here, as you can see, you have one equation here. 4 is equal to pi times 2, or beta times 2 plus pi. The question, what is pi and beta? I don't know. You have one equation, but 2 unknown parameters. There are infinite number of solutions. You plug in any pi, you get a beta. Any pi, you get a beta. So there's no unique solution. For example, here, if you have an invalid instrument, pi is not equal to 0. What if pi is 2? Then beta is 1. What if pi is minus 2? Beta is 3. So you can have so many solutions. There's no unique solution for beta. So beta can be anything in this data set. Of course, in reality, beta cannot be something, cannot be anything. It must be something. It must be a unique number that describes the causal effects of exposure on the outcome. So here I give you a comparison between valid versus invalid instrument, genetic instruments. Ideally, we want a valid instrument with pi is 0, and we don't want pi to be non-zero. If pi is non-zero, you'll be in trouble, okay? So how do we address this problem? How do we solve this equation? So there's some inspiration from literature. It's called the Karenina principle. You must know this, probably know this, by a Russian writer of last century, or maybe even before that, in the 19th century, called Leo Tolstoy. In the opening line of one of his books, called Anna Karenina, it says, all happy families are alike. Each unhappy family is unhappy in its own way. There's also a movie about this book. So that's the quote from Leo Tolstoy. And inspired by this quote, actually, this quote is important for our research because this quote told us what to do, how to solve this problem. And we apply this principle, Anna Karenina principle, to our research. We simply modify this quote. We say all valid instruments are alike, resemble each other. And each invalid instrument is invalid in its own way. What's the implication of this principle? This principle can tell us how to solve the problem when you have multiple genetic variants, okay? So let's look at one toy example. If this example is clear to everybody, you are clear about the key idea. So this table is critically important. It's a toy example, but it tells us the key idea. Suppose you have a 6 SNPs, SNP 1 to 6. In the data set, we have SNP to exposure. Think about exposure as protein. We have that number. So those are the numbers that describe the association from SNP to exposure. So if you have one more minor allele or risk allele, your exposure will increase by 0.5, 0.4, 0.7, okay? So this second column tells you the effect size of increase your SNP by 1 unit. What's gonna be the increment to exposure? Also in your data, you have SNP to outcome association. That's the third column numbers. We do a simple math, we take the ratio. So the ratio will be SNP to outcome in the numerator and SNP to exposure in the denominator. This ratio is easy to compute. You just compute it, right? Is that clear to everybody? Now, here's my question for you. Based on this ratio estimate, as you can observe, the first three rows, they give you identical ratio estimate. It's 2, 2, 2. That the ratio estimate is supposed to be an estimate of the causal effects, okay? And the last three is SNPs give you different numbers, negative 1.5, 0.5, and 1.5. They give you different numbers. So the question is, based on this data set, based on your calculation, what do you think? Which SNP is valid? Remember the principle? All happy families are alike. All valid instruments are alike. So the answer is kind of obvious. The first three instruments, they give you identical ratio estimates. So they are alike. So they should be valid instruments. And the left three, 4, 5, 6, they give you different estimates. So they are invalid in different ways. So they are not valid instruments. Make sense? It's like you give a mathematical question to 10 students. You ask them to produce a number. You observe 5 of them give you identical numbers. The other 5 give you very different numbers. What do you think? Right? Those 5 students give you identical numbers are likely to give you the correct answer. So correct answers should be the same. And wrong answers should be wrong in different ways. If you observe 2 wrong answers are identical, you might suspect what's going on with the 2 students. So that's the idea behind this. When you have multiple genetic instruments, you can actually figure out which one is valid and which one is invalid. And that's our key idea, okay? Now, the next step is how do we implement this idea in a very complicated algorithm so we can apply the algorithm to the genome-wide data. So here, next few slides gonna be slightly technical. I try to minimize it, but I have to talk about this because I do statistics. I cannot talk without using statistics. So we call it method MRSPI. I'll tell you why we call it. So the key idea is to select valid genetic instruments from the whole genome. You have millions of SNPs. How do we select them? To select the good ones. So MRSPI stands for select valid genetic instruments, select. And then after selection, how do we perform so-called post-selection statistical inference based on so-called 2-sample genome-wide association study sample statistics. 2-sample means your data from on protein and the data on the disease outcome can come from 2 different samples. That's okay. People can do this. So the key idea again is to apply the Karenina principle. So the valid instruments will form the largest group and should give you similar ratio estimates. That's the principle. Here's a cartoon here. Suppose you have 2 SNPs, SNP1 and SNP2. The horizontal axis represents SNP exposure association, and the vertical axis represents SNP outcome association. And the ratio is actually equal to the slope. So you have the slope here, right? This slope represents the causal effects because SNP outcome divided by SNP exposure, that's equal to the slope, right? So this ratio estimate of SNP1, that's a slope is equal to your causal effect. So how large is the slope? Tells you how large is your causal effects. So if you believe SNP1 is valid, you look at the SNP2. What's the distance from to this slope? So this distance from here to here, this distance is violation bias. That's your pi. That's the bias. Okay. Of course, if we assume SNP2 is valid, we can also get a bias for SNP1. If they lie on the same line, they have the same slope, then no bias, okay? Now, when you have millions of SNPs, you want to first filter out strong SNPs. So the first step is to select very strong SNPs. Those SNPs are highly correlated with your exposure. For example, protein. Those are called strong signals or PQT. So we use p-value threshold. When the p-value is highly significant, we pick them, okay? Like in genome-wide association studies, you always pick the top significant SNPs that are strongly associated with protein, okay? So that's it. We cut off, here is a cutoff line, the green line. On your right, we will be selected. On your left, they are not significant, so we don't need them. Make sense? So the first step, we pick these 6 SNPs. For example, those SNPs are highly correlated with the exposure. The next step is called a voting step. It's like Congress, right? Vote. So if you vote, the same party typically will agree with each other. And here is the idea. So we have 6 SNPs. All the rest, 5 SNPs will vote for SNP1. Are you a valid one? Okay, think about the mathematical exam. We have 10 students. So assume SNP1 is valid. We can calculate this ratio estimate, the slope. And all the other SNP will vote for SNP1. Are you valid? Yes or no? Vote for you. Okay, so SNP1 will receive votes from the other SNPs. So SNP2 and 3, they are very close to this line. So they are similar to SNP1. So similar means they are friends. I vote for my friend. That's the idea. And the SNP is 4, 5, 6. So those points, their slope is far away from this blue line. So different, I don't vote for you. Okay? So it's like a SNP 1, 2, 3, they belong to the same party. So we use this idea. So we keep voting for each SNP, which SNP. So eventually, we construct a voting matrix. I will quickly go through this here. We construct a voting matrix, and we found a party here. SNP 1, 2, 3, they vote for each other. I vote for you, you vote for me. They vote for each other to be valid. I believe you are valid. You believe I'm valid. So they mutually believe each other to be valid. And we call this party as our valid instruments. Make sense? Okay, once you believe the 3 SNPs, they are valid, they vote for each other to be valid, and we can fit a line passing the origin. And this green line, the slope of this green line is our final causal effects estimate. That's the beta we are looking for. Eventually, we get a beta. And the slope, how steep it is measures how strong is the causal effects of exposure on outcome. So here, this green line, the slope tells you the beta, which is the causal effects of D on Y, exposure on outcome. That's basically how we did this, okay? There's one more residual challenge we haven't addressed yet. What if people vote, their voting is wrong? I mean, people are not perfect, SNPs are not perfect. You might make mistakes, right? You vote for the wrong SNP. This is called IV selection error. So in the data, this algorithm might select some boundary case. You know, that SNP kind of sits in the middle. I vote for you, I vote for this side, I vote for both sides. So we call this SNP locally invalid. They are almost valid, but just a little bit not so valid, okay? We call them locally invalid. Those SNPs, they have very small, but still non-zero degrees of violation of assumptions. So a boundary case. So those SNPs might be selected as valid IV using our voting procedure. And that's a mistake, okay? We can make mistakes sometimes. And how do we account for such kind of mistake? That's the challenge. To address this, we propose a robust confidence interval. We all know confidence interval, right? the lower bound, upper bound, what's the temperature tomorrow? It's 10 to 90. That's the interval. Despite it's quite a big interval, but that's an interval. So if you have this locally invalid IVs, you might make a mistake when you select valid instrument. You might just select the wrong ones. When you make that kind of mistake, your standard confidence interval cannot cover the true causal effects. Your confidence interval may miss the true causal effects. So typically we require confidence interval to be 95%, right? Means 95% of the time, the interval will cover the true number. But when you have errors, you cannot cover with 95% of the chance, maybe only 70%. So in this example, you can see you have one locally invalid SNPs here. You mistakenly select this one into your set. And you construct a confidence interval using standard procedure. This interval here cannot cover the true causal effects, the star here. You simply miss that. Because this SNP file, which is invalid, will drag your confidence interval to this side. So you cannot cover the true causal effects. So your standard CR will fail due to your mistake. So our solution is to construct a robust confidence interval using a new method called searching and sampling. It's slightly technical. I will quickly go through it. So it's based on Monte Carlo sampling. So we sample from our estimates multiple times, typically 1,000 times. So n from 1 to capital N. Capital M typically is equal to 1,000. We just do sampling using computer. So computer can do this kind of Monte Carlo sampling for you. And we try to find one particular sample number which is very close to the true number. So in this cartoon, I can show you, your estimated number is here, the blue one. You sample from this blue one, sample circle, and the true value, this star, purple one. You keep sampling. When you have a lot of samples, eventually you're gonna find someone which is closer to the truth, okay? You keep exploring. Eventually, you're gonna be very close to the truth. And this triangle is the number we try to find, which is very close to the true value. And to implement this intuition, what we do is like this. We calculate the ratio estimate in each sampling. And for each sampling, we try to construct a pseudo interval. It's not really the true interval, but we construct a pseudo interval. So we still use a voting procedure. So for each number in this pseudo interval, more than half of the instruments will vote for this number to be the true causal effects. So more than half believe that's true, okay? We call it majority rule. More than half agree this is a true number, okay? And we just aggregate all such kind of pseudo intervals to construct a robust interval. So keep one sampling, two sampling until a certain sampling, and we take the lower bound and upper bound. We construct a very big confidence interval. So this is the true causal effects in like star here. We're gonna cover it, okay? This interval is guaranteed to cover these true causal effects with 95% coverage level. And that's the interval we're gonna use if you suspect your IV selection is not perfect. If you suspect you made a mistake, here is the rescue. We still can rescue you if you make a mistake, okay?
There's a question in,
yes.
in the chat. So this method relies on a large number of
SNPs.
instruments.
Yes.
And the question is, what do you do in situations where you want to do analysis with a small number of SNPs, like 1 or 2?
Well, 2 is not feasible.
What would be the number?
If you have 1 or 2 SNPs, this method won't work. At least 5, 6, or 10 or more. Have 2, won't work. You have 2 SNPs. I think you are wrong. You think I'm wrong. You need to vote, right? 3 maybe work. Maybe 2 versus 1. There's 3 ways. You have 2 students. The number here is 2. There is 3. I don't know which one is correct. So I would say more than 3, including 3. This this method in principle works. 2 more, 1 more work. Okay, yes. Now we apply the methods to the plasma protein data. We try to find any plasma proteins associated with Alzheimer's disease. So plasma proteins, they have non-zero causal effects on Alzheimer's disease, and we use genetic variants as instruments. So we analyzed 912 plasma proteins in 54,000 people from UK Biobank. And this paper, actually, not my paper, this paper from UK Biobank was published recently in Nature. And we also analyze data from Alzheimer's disease here and using our methods. So we found 7 proteins, plasma proteins. They are significantly associated with Alzheimer's disease using our method called MRPI. Some proteins, they seem to be protective, and some proteins seem to be like increasing the risk of Alzheimer's disease. This plot is called a volcano plot, and the green ones means they have positive effects. The red ones means they have negative effects. Negative effects means they reduce the risk. Positive effects means they increase the risk of Alzheimer's disease. And the horizontal axis represent the estimated effects on Alzheimer's disease on the log odds ratio scale, okay? And the vertical axis represent the p-value on the minus log base 10 scale. So this one, this TREM2 is highly significant, okay? P-value is smaller than 10 to the minus 15, highly significant, and also seems to be protective because it's on the left-hand side, it's negative. The green ones are positive, okay? So we found those 7 proteins in our data. And we did some sensitivity analysis by including a few other popular methods in the field. I think we include 7 other methods, try to see how sensitive the results are if we apply different methods. If all the methods give you similar results, we have a higher confidence our data analysis results are more likely to be true. If you see any differences across different methods, you might be less confident. So we observe all those methods, they tend to give you similar results, slightly different numbers, but the conclusions stay almost the same, okay? So that's a good sign. So there are some previous studies on proteins and Alzheimer's disease. For example, this paper says this TREM2 proteins and the CD33 proteins, they are involved in the microglia pathology in Alzheimer's disease, and this paper on TREM protein. So we did some so-called enrichment analysis. We found those proteins, the genes encoding for those 7 proteins, they are overly expressed in the brain tissue, microglial tissues, macrophage, and other tissues. We also did a so-called gene ontology enrichment analysis by including those 7 proteins we found, and go through this gene ontology database. And we found a lot of biological process or functions that are enriched. For example, this MHC class 1 protein binding, which is important in the adaptive immune process. Now, I have about 10 minutes left, so I will spend like 5 to 6 minutes on AI, AlphaFold. So AlphaFold is a powerful tool to predict the 3D structures of proteins, so the protein binding problem. And protein is a chain of amino acids. Once you have a chain of amino acids, they bind in the 3D space into certain structure. And this AlphaFold uses deep neural networks published in 2021 in Nature. They claim they provide a solution to a 50-year-old grand challenge in biology. So here is the basic workflow of this AlphaFold2. The input of this AI tool is a sequence of amino acids. amino acids long sequence. You provide the sequence, this AI, AlphaFold2 AI tool can give you a 3D structure, okay? The 3D coordinates of each amino acid in a 3D space. That's how it works. And in the middle, they are so-called AlphaFold based on evolution theory. AlphaFold is like a transformer. It's a 48 blocks of neural networks, deep neural networks. It did a lot of calculations. Eventually, they figure out the coordinates of your amino acids of this long protein sequence, okay? And for our 7 proteins, we look at the missense genetic variation. Some people, for example, this is SNP, RS2455069. Some people located in chromosome 19. Some people with A allele, some people carry G allele, okay? Maybe I carry G allele and you carry A allele. Different people may carry different alleles, okay? And if you have A allele, that's your amino acid here. And you have G allele, that's your amino acid here. 3 base pairs is a codon, right? So AGG is a codon for this amino acid, and then GGG is a codon for glycine, this amino acid. If you have a different amino acid, your 3D structure may be different, as highlighted here. If you have this amino acid, that's a local structure. You have G allele, that's the yellow part, this is a local structure. And then you can see there is a local structural changes if you have different amino acids due to missense genetic variations, okay? That's the idea. And this genetic variation is used as our valid instruments, okay? So we select this SNP as instrument. So I just show you some other 3D structures of other 7 proteins. Okay, so last part is about drug development. When you have these 3D structures, you can design chemicals, to bind to those proteins. That's beyond my knowledge. I'm just a high level say that's possible to design some compound to reduce number of some bad proteins or increase the level of good proteins, so you can cure or at least to some extent of your disease. Also, those proteins also provide targets for drug repurposing. For example, we search the proteins in drug databases. I provide 2 drug databases here. You search CD33, which is a protein we found in the data. And actually, this protein has been targeted by existing drugs. For example, this drug from this company has been approved by FDA for treating leukemia. So this protein we found is related to Alzheimer's disease, but this protein is also targeted by this drug for treating acute myeloid leukemia. Does that mean we can repurpose this drug for treating Alzheimer's disease? That's a hypothesis. I don't know, but that's requires further research. Another example is this protein we found called RET, and there exist 2 drugs approved as RET inhibitors for the treatment of non-small cell lung cancers. Does that mean these 2 drugs can be repurposed for Alzheimer's disease? That requires further research. Okay, so the proteins provide drug repurposing opportunities. To conclude, we developed a method called MRPI, which can select valid genetic instruments for causal inference to identify putative causal biomarkers for the disease outcome of interest. And we use AlphaFold2 to predict the 3D structural alterations due to missense genetic variations selected as our instruments, okay? And we develop an analysis pipeline for drug target discovery and the possible drug repurposing. And the software is public available, freely available in GitHub. For future work, we will try to develop a shorter confidence interval to improve power so we can detect more biomarkers. And we are trying to analyze other type of omics data, for example, iron-seq, isolation, and metabolites. And also analyze data from other populations of non-European ancestry. So far, the data is from European population and may not be generalizable to other populations if there is heterogeneity across populations. And next step, we try to use AlphaMissense, another AI tool, to do so-called functional predictions of missense genetic variations. Last, but of course not the least, this is probably one of the most important slides. I want to thank the wonderful collaborators who are involved in this project. The first one is Min Hao, is still a student in Hong Kong. I used to supervise in Hong Kong. And Gary Miller from Mailman, and Badri from Department of Neurology here at Columbia, and Andrea Baccarelli, still here now, but in 2 weeks, he will be the Dean of Harvard Chan School of Public Health starting January 1st. And Zhijian from Rutgers University Department of Statistics. And you are welcome to follow me on Twitter. I'm not very active, but I still check Twitter a few, every 2 days, and also my lab page at Columbia. Thank you so much. If you have any questions, I'm happy to answer.
Yeah, thanks. So this was the most clear explanation of Mendelian randomization.
Thank you.
Approach that I've heard. So congratulations, this was amazing.
Thank you.
So we're open to questions.
In chat? I cannot see from my computer. But...
could you please comment on LD relationship between selected genetic instrument, for example, what kind of
right. Yeah, that's a very good question. We didn't mention because that's probably too detailed question. So in the algorithm, we first need to do LD clumping to select independent SNPs. Typically, the LD score R squared is below than 0.1 or sometimes smaller than 0.1. So to make sure we have independent SNPs, the SNPs we're going to use are not in LD.
So kind of a related question. So, a lot of proteins have cis-pQTL signals.
We use the cis ones.
Yeah, so the question is whether you use cis and trans or just cis.
Just cis.
And then if you're using cis, then you're applying the pruning procedure to select the instruments, right?
Yeah, only to cis.
So can you comment on the number of instruments that you end up using?
Actually below the number of instruments eventually used for protein varies. Eventually in the analysis, probably 5 to 10. Not many. After you did all those sections, in the end, we use 5 to 10.
What if you just have one haplotype that leads to a major difference in protein concentration? So you have like one instrument, you probably can't apply your instrument validation procedure.
Yeah, that's true. Yeah. That's probably one limitation of this this field. Actually, it's very hard to do. We probably need to do more research, future work. Yeah. Any more questions?
Yeah, maybe I can follow up with another question.
Okay.
Kind of interested in this topic. How often do you see violations of the horizontal? How often do you see more evidence for horizontal?
Yeah.
In terms of like how many SNPs do you really need to drop off the analysis on average across the proteome?
That's a good question. It varies. Some SNPs, they are highly pleiotropic, like ApoE, some SNP in ApoE gene. That's just related to many traits. Some SNPs, they are just not so pleiotropic. I don't have an exact number for like what's the proportion will be pleiotropic, but there are a good number of SNPs that are filtered out because they are likely to be pleiotropic.
And how do you handle the HLA area, for example, or this extended?
Yeah, we don't use that part.
You don't.
That part is a highly polymorphic. Yeah, and yeah, we don't use that part.
Great. And other questions?
Hopefully it's clear, right? So
Very clear.
Well, thank you.
So much. This is
Yeah.
amazing.
thank you so much for inviting me.